Marching onwards, part 2 today, which is video part 3, but outline part 2 is memory and learning. The reason we're talking about memory and learning in our class on synapses is that memory and learning is all about changes to synapses. The way you learn things is by physically changing the synapses in your brain. This process actually starts during nervous system development, so the macro organization of the nervous system is laid down during embryonic development. The brain, the spinal cord, the major parts of the brain, all that stuff is set up during embryonic development, and it's very tightly regulated by gene expression and signal transduction. But what we're going to focus on here is how the brain develops and remodels after birth, because obviously you continue learning throughout your life, and that process involves changes to your brain. The first major thing we'll talk about related to this is neuronal plasticity, which is a bit of a review. We've talked about this before. Neuronal plasticity is how your brain is going to change the synapses, the physical connections between neurons in response to activity. Basically, there is a competition that goes on between all the neurons in your brain for things called growth-supporting factors. And in order to maintain things like synaptic connections, you need growth-supporting factors. Well, if a neuron is being used a lot, then it's getting a lot of those growth-supporting factors. And if it's not being used a lot, it's not getting those factors. This means that your brain is basically only keeping the synapses that it needs. And this makes sense from a perspective of like managing energy use in your body, because synapses are complicated things and they require a lot of energy. So if you have synapses that are sitting there, you're maintaining things like vesicles full of neurotransmitters, you've got ligand-gated ion channels, the receptors. All that stuff costs energy to make and maintain, and if you don't need it, then get rid of it and save the energy, put it towards something you're actually using, because energy within an organism is a limited resource. So what we see is that only the synapses that are in use are maintained. And we can see that in this figure, which you have seen before, where here, neuron number 1 and 1 has an active synapse, and over time you add connections due to that prolonged use. And down here, neuron 2 is not being used, so over time, that synapse is lost. That gain and loss of connections is called neuronal plasticity, and it's typically in response to activity. We can also consider the strength of synapses. You can have a connection, and that connection can be very robust and make it very easy to transmit a signal to the next neuron, or it can be very weak and it's difficult to transmit a signal to the next neuron. That strength can also be modified. So here we have an example where two synapses are active together over some prolonged period of time. That's going to result in both of them being strengthened. And by the same token, if they are not used, then the strength of the signal from either of those synapses will weaken, and that's different and independent from that synapse being lost entirely, like we saw on the previous slide. The big idea here is that memory is a use it or lose it system. If you have information that you want to remember, the way to remember that information is to use that information repeatedly every day. Use it a little bit, and that's going to tell your brain to hang on to it. That's why I tell everybody, give me 20 minutes of bio practice every day. That tells your brain, this is important, make it readily available. What we want to do here is connect the changes we just talked about to memory and learning. Neuronal plasticity is like the big thing here. This is a bit of a review, but there are different types of memory in the brain. We're going to talk about two right now. There's short-term memory, or STM. This is where information is held for a very short time and then it's released. The two parts of the brain that are involved in short-term memory are the cerebral cortex, so the outer surface, and the hippocampus. And the important thing here is that short-term memories that are in your short-term memory, are accessed via the hippocampus using temporary links. And I've used the analogy before that your short-term memory is like RAM on a computer. You can put whatever you want in there, but then if you need to do something else, you're going to replace it. So the hippocampus can store some number of items. In psych, you've probably heard 7 plus or minus 2, 5 to 7, 5 to 10, somewhere around there. Those are going to be used through the hippocampus, but then once you either don't use that information for some amount of time or you read or engage with some other information, then it's going to be replaced with whatever that new stuff is. So short-term memory is important for acquiring memories, but it is not useful for maintaining memories. For memory maintenance, for keeping things, we need to use long-term memory, or LTM. This does not involve the hippocampus. This is where you make permanent connections in the cerebral cortex. So the cool thing about long-term memory is that as far as we can tell, it doesn't have an expiration date. Stuff that gets into your long-term memory is basically there indefinitely. And in terms of the capacity, again, as far as we can tell, it's basically the same. It's more or less unlimited. Those are both very difficult things to test experimentally. So put that little disclaimer on there that it's as far as we know. But as far as we know, long-term memory, which again uses the cerebral cortex and permanent connections, that is going to be indefinite and it is infinite in its capacity. These topics are connected to something very important for general biology as a course, and that is, of course, long-term potentiation, or LTP. And we've talked about this before. This slide is actually a review. You've seen this flowchart before, so we're not going to go through it step by step. But you know that long-term potentiation is a lasting increase in the strength of synaptic transmission, and this is really important for both memory storage and learning because it's a process that reinforces both the retention of memories and your ability to quickly access those memories or retrieve them. We can see in this part of this flowchart how long-term potentiation plays a role in memory formation and retention. When you have something in your short-term memory and it is stored in your long-term memory, that is called encoding. But then when you need to use it again, you need to retrieve it back into your short-term memory. That loop triggers long-term potentiation, which reinforces both the encoding and retrieval sides of that process. So you're basically inducing a positive feedback loop of increasing retention and increasing your ability to retrieve that information when you need it. That's what's going on with long-term potentiation. Now that's as far as we've gotten for LTP in the past, but today we're going to go a step further and talk about the mechanisms. In order to induce long-term potentiations, you need to meet two conditions. First, there needs to be a high-frequency series of action potentials being sent to the postsynaptic cell. So basically, for the synapse in question that you're going to strengthen, you need to have repeated signaling. In other words, you need to have temporal summation at that synapse. You also need depolarization of that postsynaptic membrane from a different stimulus, from a different synapse at the same time. In other words, you also need spatial summation. So what two things need to happen in order to induce long-term potentiation for a specific synapse? You need both temporal and spatial summation. If you meet these two conditions, you strengthen the synapses that are working together. So this figure I showed you a few minutes ago, this is actually showing kind of schematically the process of long-term potentiation. These two neurons are firing frequently and together, and over time that strengthens the signal from both, strengthening that synaptic connection and therefore increasing the strength of that signal is called long-term potentiation. So now we're really going to get into the weeds, and I will tell you, I want you to be able to describe this process, but you're not going to need to know like the specific names of the ion channels. We're going to see them in the next figure I'm going to show you on the next slide, but you're not going to have to know like which ones are which. So how does all this work? Well, in the postsynaptic neuron, there are two types of ligand-gated ion channels. Before LTP, so when a synapse is relatively weak, it's not a robust connection, one of those is in the membrane, so it's a transmembrane protein, and it is available to be stimulated by neurotransmitters. But the other one is in storage, kind of below the plasma membrane. It's not actually being used. That means you're not going to trigger a depolarization that's large enough to hit the threshold based on that single synapse. You need to depolarize from a different source in order to reach that threshold potential. In other words, what needs to happen in order to reach the threshold is spatial summation. That's before LTP. After you've induced LTP, both of those types of receptors are going to be in the membrane, which means you can have a stronger depolarization from that single source, which means you're more likely to trigger an action potential from that one synaptic connection. We're now going to look at this graphically and see kind of the steps in the process. The thing I want you to get here is the basic idea of what a pre-long-term potentiation synapse looks like compared to a post-long-term potentiation synapse. First, our before situation. We have an action potential arrives at our presynaptic neuron. It triggers the release of a neurotransmitter, in this case glutamate, but I'm not going to ask you that. And in this postsynaptic cell, we have two types of receptors. There are purple ones and there are pink ones. You don't need to know the names for each of these, but I'm going to use the names right here in the text so that you can relate the text to the figure easily, but you're not responsible for the names. So initially, you have NMDA receptors. The NMDA receptors are going to be opened by that neurotransmitter, but they are blocked by magnesium ions. So even when they're open, you still can't trigger a depolarization in the postsynaptic membrane, and that causes no action potential as a result. So you're not transmitting the signal effectively to the postsynaptic cell. The second type of receptor, the AMPA receptors, the pink ones, they're not even being used. They're in storage. They're not actually in the membrane being used right here. Now, as you're practicing, as you're developing this synapse, as you're using the information associated with this synapse and connecting it with other things, you're causing a depolarization from a different synapse for this same postsynaptic cell. So pretend if this figure was to be wider, you would see another synapse active, over here or something. I don't know if you can see my finger in the thing, but be over here, you have another active synapse going. That triggers a depolarization and it diffuses along that plasma membrane. So the depolarization from pretend there's another axon over here. So that depolarization travels, because those ions diffuse, it travels over here. When this membrane is depolarized a little bit, two things are going to happen. One is the depolarization triggers the unblocking of those purple receptors. So when the inside of the cell depolarizes, it forces that magnesium ion away from that receptor. Once that happens, sodium and calcium can flow into this postsynaptic neuron through the now open purple receptors here. This depolarization also causes the AMPA receptors to come out of storage and be actively placed in the membrane. So over time, when you're using one synapse in conjunction with another consistently over a period of time, you're increasing the number of receptors that are present in that synapse. Finally, once we have induced long-term potentiation, we have lots of receptors, both the pink ones and the purple ones. Now remember, at our resting state for this postsynaptic membrane, those purple receptors are going to be blocked by an ion, a magnesium ion. So even if you trigger them with the neurotransmitter, you're still not going to depolarize. The AMPA receptors, the pink ones, do not have that problem. So when an action potential triggers this synapse right here, initially what's going to happen is you trigger the pink receptors, you have an initial depolarization, and then when a subsequent action potential comes down this presynaptic neuron, the purple receptors are then going to be unblocked because the initial depolarization resulted in the kind of kicking out of that magnesium ion. So then on the second try, you can use all of the receptors in the membrane, and this is only showing 3, but it would actually obviously be thousands, if not more. And you're using all of these receptors at once, and that second wave, that might be sufficient to trigger an action potential. So let's go through this one more time. I know that was a lot, but I just want to compare the pre-LTP situation to the post-LTP situation. Before LTP is induced, there's only one type of receptor in the membrane, and it is blocked by an ion. So activating this synapse is insufficient to trigger an action potential. But when you have spatial summation involving this synapse and then another synapse somewhere else on the same postsynaptic cell, that is going to allow this region to depolarize, which causes those second types of receptors, those pink receptors, to be used. And you have to do this a lot in order to get a lot of those receptors in that membrane. That's the practice you're doing when you're trying to remember something. This is spatial summation. What we're seeing here is the effects of spatial summation. This synapse alone is not going to do anything, but when you combine its activity with a different synapse somewhere else, now you can have an effect. After some time of that process happening, you generate a situation like this, where you have lots of receptors. Some of them are going to be blocked by magnesium, some of them are not. And in order for this to work, you need temporal summation. So you need to have a signal, an initial action potential, triggering this synapse. That's going to cause an initial depolarization using the pink receptors. That's not going to be enough to trigger an action potential, but it does unblock the purple receptors. Then you have a second wave come down. As long as that hits before the first one dissipates, now you're going to use all of the receptors on that second wave, and that's going to trigger an action potential. So that's your temporal summation over here. So that's why both spatial and temporal summation are required for this process. And that's how we generate long-term potentiation. Again, I don't want you to know like the specific neurotransmitters or the names of the specific receptors. I just need you to know the basics that initially you have one type of receptor that's not working very well. Spatial summation triggers the addition of a second type of receptor, and then once two types of receptors are present at a synapse, temporal summation allows you to trigger an action potential with that single synapse alone. That's when you have long-term potentiation. You've heard me say this before, but this is the point of what we do in Gen Bio. This is why this is an active learning course. The things that you do in workshop promote long-term potentiation. If you're just reading the textbook or reading your notes, that is a single stimulus. You're just reading. That's not going to promote long-term potentiation because you need spatial summation. But if you are working with the material, you're doing the sequencing, you're consulting your notes, you're talking out loud, those are multiple stimuli and therefore multiple neurons, multiple synapses being activated at once. So you are triggering spatial summation of a bunch of different signals, and in doing so, it becomes mutually reinforcing. The repeated use and repeated active use, where you're doing multiple things at once, that tells your brain, this is important, I need to be able to access this when I need it. And the goal here is to get to the exam and read a question, and the stimulus of that question is going to trigger the pre-existing networks in your brain that you have strengthened by doing this kind of practice. And that's why you should never change an answer on a multiple choice exam. When you read a question and you immediately know an answer, that is your long-term potentiation doing the work for you. But if you go through the rest of the exam, you go back and you kind of think about other stuff, what you're basically doing is overriding that long-term potentiation. You're overriding that training that you've done in your brain. So this is not just a Gen Bio thing. This is an every class thing. Don't change answers on multiple choice exams because you're basically kind of outthinking your own brain, and you're working against the training that you've done. So to summarize synapses, these are connections between neurons. They are electrical or chemical. We only talked about chemical today. Most of the synapses in your body are chemical synapses. There are many different inputs that are going to lead to a specific effect, and these junctions play an important role in memory and learning and long-term potentiation. Now, this is not the end of this series of this class, lecture series 20. We still have to talk about senses and sensory reception, but this first half is all about synapses and how they're involved in memory, and we're now done with that.